survive and exist and thrive in these environments means that all of the associated organisms, the whole community of organisms, is present in the caves or in the volcanoes or in the deserts, which means that not just the organisms themselves, but also their viruses are present. And in fact, it may be that viruses are the way to communicate between organisms. Viruses carry genetic material, and some of those genes actually provide capabilities to organisms that the viruses infect, that organisms without the virus infection wouldn't be able to exhibit.
The science packages that are on some of the missions in the future and in the recent past, they were designed to search for certain types of life and things. Can you sort of touch on why they were designed that way? How would they even guess before the thing launched what to design for?
So when we design life detection experiments for other planets, we have to consider what kind of life we expect to find. For the Viking mission, that was the first mission to send life detection instruments to Mars, we didn't know about all this extreme life. So the Viking life detection package was designed to find organisms that are living in environments similar to the environments that humans consider hospitable. When the Viking mission was launched to Mars, we didn't know about hydrothermal vents and the organisms that live at the bottom of the ocean. So those life detection experiments were trying to find both the components of life, the organic compounds in the mass spectrometry experiment, and also trying to look for reactions that would indicate life in the labeled release experiment. The labeled release experiment on Viking actually did observe reactions that released labeled carbon-14. So we did see a signal from that experiment, but that signal is consistent either with organisms being present on Mars or with reactive compounds to be present in the soil of Mars. Subsequently, we've discovered that there is in fact perchlorate compounds in the soil of Mars. So that explains the signal that we saw during Viking from the life detection labeled release experiment. For the mass spectrometry experiment on Viking, it was observed that there was methane with chlorine compounds around it, chlorinated methane. Instead of just carbon and hydrogen, which is the normal methane, some of those hydrogens were replaced by chlorine. This is consistent with the cleaning fluid that was used to clean the life detection experiments on Viking before launch. So it may be, and it was interpreted at the time, that signal was thought to indicate that there was cleaning fluid left over from before launch. A noise problem, a contamination event, an indication of Earth life as opposed to Mars life. More recently, those experiments have been revisited with the understanding that there is in fact perchlorates present in the soil of Mars, and that signal may actually have indicated the perchlorate reacting with organic compounds that are present in the soil of Mars. So this illustrates both the challenges for planetary protection of allowing contamination to get into your instruments, but it also illustrates the difficulty of interpreting life detection experiments that we do in places that we send our spacecraft, because it's just a really hard problem, and no matter what experiments we send, you can't necessarily interpret the results effectively if the results are not what you expected up front. So expecting Earth life to be like human life that we find on Mars, we got a signal that was just not easy to interpret.
I said I was an optimist driving up here, and I'm just going to go on a limb and say, yes, we're going to find life on possibly Ganymede in Europa, and the tests are going to come through and we're all going to cheer. Is there going to be a big rush to bring this life form back to Earth, and that involves your department? So
if we were to find life on Europa, identify signals of life on Europa, identify signals of life on Enceladus or Mars, it is likely that humans would want to study that life further. The best way to study life is to get it back in a laboratory on Earth and investigate all of the characteristics using the instrumentation that we have available on Earth, which is the best instrumentation. We have much better instruments in the labs than we can possibly put on a spacecraft. In order to bring back samples that might have life, samples from Europa, samples from Enceladus, or samples from Mars, we have guidelines already developed by the international community that describe what kind of processes do we need to perform to ensure the safety of the Earth as we bring these samples back. And so essentially, those requirements involve containing the samples to the same level that we would contain hazardous materials from Earth, Ebola virus or smallpox, Ebola virus or smallpox. We have procedures and we have containment facilities, a biosafety level 4 containment processes and procedures that we use to contain hazardous organisms on Earth. So when we bring samples back from another planet that might have life, we will apply those same procedures, not because we have confidence that the samples are hazardous, because we don't. We don't even know if they have life, but because of the potential for life and our lack of understanding of what those organisms might do when we release them, what those organisms might do if they were released into the Earth's environment. So we will not release them. We will bring them back and study them under containment until we have confidence that they are not hazardous. And after that point, we might let them out of containment. Recently, the European Science Foundation has produced a study that identifies viruses as being Recently, the European Science Foundation has produced a study that has identified viruses as being of concern. Viruses are much smaller than the organisms that we have previously thought would be needing to be contained. But if there are viruses that use the similar type of genetic material that we have in organisms on Earth, we would definitely want to make sure that those viruses cannot get out of the containment, as well as any organisms that might be present in the samples.
When the general public hears the word virus, they always fear and panic because of what known viruses, smallpox, and these different types that have killed people. Yet in recent years, Dr. Stedman and other people in Germany, they've taken viruses from these extreme environments that we are talking about and have genetically altered them and have used them to cure cancer and HIV. Possibly whatever we find on another planet could be beneficial. I'm trying to get you to talk about the benefits of viruses.
So when people hear the word viruses, they think, oh my goodness, it's a disease. And it's true that there are certain viruses, like Ebola and smallpox, that are hazardous to humans. But there are viruses in your drinking water, there are viruses in your bath water, there are viruses in the rocks around us that don't infect humans. There are viruses associated with the microbes, with the biology that is present in the environments that we all are in. So most viruses are totally harmless. In some cases, in fact, medical science has been able to take viruses that do infect humans, or perhaps cause warts, or perhaps just coexist with humans and don't really cause disease, and use them to alter the physiology of human beings and potentially cure diseases by providing additional genetic material that might enable humans to regain capabilities that might be caused by a genetic defect. I can say that over if you want me to.
Yeah, a little bit. Just that one.
Yeah. So recently, medical science has been able to use viruses that are associated with humans to actually introduce additional genetic material and cure diseases that might be caused by defects in the genes of the person who doesn't have them.
I think we did it.
Okay.
We covered it
Anything
upstairs,
else?
We've covered it down here. I don't want to beat a dead horse by having you
Okay.
say it in 5 different ways.
Okay.
I think we got it.
That's it?
Yeah,
Okay, cool.
that was amazing.